Hello everyone, this is CJ Zhou from Taipei Veteran General Hospital, and I am very honored to be here to share my recent study about combining the nanotechnology and the CRISPR-Cas9 gene editing tools for treatment with the genetic disorder syndromes. In recent years, the main topic in biology or medicine that has been discussed the most is the gene editing. The deliberate alternation of the state DNA sequence in living cells. The human genome editing has many potential therapeutic applications and anticipated to be among the most important areas of the biomedical innovation in the next 5 years. Here, I would like to introduce a potential non-viral therapeutic approach for inherited disease by combining the supramolecular nanoparticles with the CRISPR and the Cas9 gene editing strategy. And in the beginning, I would like to introduce myself. My name is CJ Zhou, and I am an assistant research fellow at the Department of Medical Research, Taipei Veteran General Hospital. My research focuses on human pluripotent stem cell biology, the reprogramming, the gene editing, and the nanotechnology. And this is the outline of my presentation today. And first of all, I will introduce what is the inherited disease and the treatment strategy. The inherited disease are a general group of the genetic disorder that's caused by one or more abnormality in the genome that may affect daily body functions or cause life-threatening problems. To the date, scientists are still exploring ways to treat this inherited disease. And the treatment strategy, previous to the gene editing strategy introduced in clinical base, the available treatment for the genetic disease is only cell drug therapy. However, since the medication can only reveal the syndrome and hence the patient needed to take the drug for the entire life and needed to pay high medical expenditure. Thus, exploration in gene therapy is getting high attention from scientists as he can identify the mutant genes and remove and replace with the functional gene. By using the gene editing strategy, the patient will be fully recovered from the disease and be able to be free from the long-term medication side effect. And when we're talking about the genome editing, we must not exclude out the two scientists with the Dr. Jennifer and the Dr. Emil, but they had achieved one of the most stunning breakthroughs in the history of biology, becoming the first to use the strategy called the CRISPR and the Cas9, alter the genetic makeup of a living organism. Their gene editing tools will allow the scientists to efficiently insert or delete specific base of the DNA with the unprecedented precision. And next, I will talking about the virus and the non-viral vectors. The delivery of vectors are required to introduce the CRISPR and the Cas9 into the cell. Currently, there are two types of the delivery vectors, which are the viral vectors and the non-viral vectors. The viral vectors like the retrovirus, lentivirus, and the AAV were the common delivery vectors that are fairly successful in terms of gene editing. However, by using the viral vectors, we encounter the immune problem, the insertional mutagenesis, the small packaging capacity, and the off-target side effect. It appears that the viral vector may not be a safe vehicle for patients. Hence, another revolutionary vehicle like the non-viral vectors is urgently needed. I would like to emphasize the non-viral delivery vectors, seeing as they are non-immunogenesis and have a high packaging limit, suggesting a safer and feasible option for the patients. And let's go through the non-viral factors. And here, I will introduce the several examples of the supramolecular nanoparticle, the SMPS. Previously, we demonstrate a convenient and flexible self-assembled synthetic strategy for producing the SMMP factor by mixing the three compounds, the ADPOM, ADPEG, and the CDPI. The multi-variant of the AD and the CD molecular recognition allows modular control over the size. And in our study, we try to use the SMMP factor to encapsulate the CRISPR and the Cas9 material and deliver it into the cell for gene editing. And lastly, let's go to the combining the nanotechnology and the CRISPR gene editing. And in here, as to take the S-linked retinoschisis disease as an example. This disease is mainly caused by RS1 gene mutation, which result in function loss of the retinoschisis protein, leading to the retinal splitting and result in complete loss of the vision. Up to date, there is still not any medications or the treatment not able to completely cure this disease. Hence, the gene editing is its field that we're currently exploring and combining the nanotechnology and the CRISPR-Cas9 gene editing for treatment with the SLRS disease. And in our study, the two molecular nanoparticle, the SMP, were developed for co-delivery of the Cas9 RNA plasmid and the donor plasmid, encoding the RS1 and the GFP, enabling the CRISPR-Cas9 mediated knocking of the RS1 gene in mouse retina. After the Cas9 RNA formation in vivo, the CRISPR-Cas9 mediated knocking of the RS1 gene is carried out in two consecutive steps following the homology-independent target integration, the heating strategy. The heating was developed based on the NHEJ pathway to enable robust knocking of long-dividing cells in vivo. In brief, the heating strategy introduced the two predetermined CRISPR-Cas9 target sites into the donor DNA. After cutting the target site present in both the genomic DNA and the donor DNA, the resulting three double-strand break site undergoes endogenous DNA repair by the NHEJ pathway to achieve the gene integration. And this finding has been successfully published in the Journal of the Advanced Science. In summary, we introduced an in vivo CRISPR-Cas9 mediated knocking approach using the two SMMP factors. And apart from the SLRS disease, we also try to apply this gene editing and the SMMP techniques on hemoglobinopathy, like the sickle cell anemia. To enhance the delivery efficiency, we introduced an effective CRISPR-Cas9 mediated knocking approach using combining the SMMP and the SNSMD strategy, which could enable the step-by-step delivery of the Cas9 RNA complex. And this is the RNP system and the donor DNA encoding the HBB and the GFP genes encapsulate into the two different SMMP factors. And this finding has published in Science Advance this year. And finally, I have to thank for the two important mentors, the Professor Cho and the Professor Zheng, and my colleagues and the financial support from the most. And thanks for your attention.